In every biology class, you always start off at the cellular level. And this is especially true for pre-AP biology. And really what you do is you work your way up from there to body systems to organisms themselves. And so, thinking about this in an order, microbiology really builds off to the rest of the sciences as well, which means that to truly establish a good foundation for the entire curriculum of biology, you need to have a good understanding of microbiology at the cellular level. And one of the most important things to know at the cellular level is organelles and cells. Here I have a very rough drawing of a cell and all organelles that you really need to know for pre-AP biology. The first one is the nucleus. The nucleus. And in animal cells, it's at the very center of the cell usually, and it's the essential brain of the cell because it contains all of your genetic information, all of your genetic info. All 23 chromosomes and your entire genome is all held within this one organelle. Whatever makes you, you, is held within the nucleus. Now you might be asking yourself, what is this purple condensed part in the nucleus? Well, that's the nucleolus, And its function is to create ribosomes, which are vital in the cell and body's function. And we'll get into that later in the video, these blue dots over here. In the nucleus, many important functions are carried out, the main one being transcription, which is an important function that we can definitely get into in a later video because it's a very detailed process. And adding to that, the nucleus isn't just one membrane and one circle, but it's a double membrane layer. So you can imagine there'd be two membranes around this nucleus in this sort of shape, and it's quite porous, which means that it has channels so that the products of transcription, this process, and products of processes in the nucleus can exit to the rest of the cell. Now, we talked about ribosomes, but really, what are they? Well, these blue dots are ribosomes, and their sites, well, I'll write ribosomes first as a heading, and they're sites of protein synthesis. Synthesis. As we know, and as we've talked about in our biomolecules video, proteins are essential to the human body. They make up steroids, they make up hormones, they make up motor proteins, and really many important things in a very versatile array. And these ribosomes are the site of protein synthesis. We have two main classifications of ribosomes, those that are found in the cytosol, or this gel-like substance in between organelles, and these are called free ribosomes. And we have these ribosomes on this organelle, which we call the endoplasmic reticulum, and we call these bound ribosomes. Now, what's important to note is that free ribosomes, when they make proteins, the products or these proteins usually stay in the cytosol. So these usually stay in cytosol. But the ribosomes that are bound on the endoplasmic reticulum have more functionality, as the proteins that they create are usually sent either outside the cell or onto the cell membrane, which is where the endoplasmic reticulum really comes into play. The endoplasmic reticulum is a structure that really wraps around the nucleus, and its main purpose is to store proteins in vesicles to transport. What is a vesicle? Well, if we take the proteins from the ribosomes, and let's just make it purple, what the endoplasmic reticulum does is as the protein is leaving the endoplasmic reticulum, it'll actually be encircled in a lipid monolayer. like this, so that it's easy to transport throughout the cytosol. And when we talk about the endoplasmic reticulum and ribosomes, it's also really important to look at the functionality of the Golgi apparatus. The Golgi apparatus. Its functionality is really connected to the endoplasmic reticulum because it takes the vesicles from the endoplasmic reticulum, often abbreviated as the ER, and what it does is it modifies them and packages them to be sent outside of the cell. So it modifies and packages products from the ER, products from the ER, and it sends them outside of the cell, or onto the cell membrane. Now, when you talk about the cell membrane, we really have to ask ourselves what exactly it is. And this cell membrane is made up of a lipid bilayer. If we zoom in onto the cell membrane with a microscope, we can see that it actually looks something like this. We know the lipids, also known as phospholipids, if we refer to our biomolecules video, are found on the cell membrane. And it looks something like this, with each having two fatty acid tails. And an important thing about the cell membrane is that it's not extremely rigid. What happens is that because of the lipid's functionality, it actually allows for certain substances to pass through the cell membrane. For example, carbon dioxide is very easily diffusing through the cell membrane, while other objects, such as ions, are very hard to pass through. So, for example, a calcium ion won't pass through because it's an ion, and lipid bilayers really don't like ions. They're really selectively permeable to certain substances. And this interaction of allowing substances inside and outside is extremely vital to the cell's health. What's important to note is that out of these multitudes of organelles, this really section right here of ribosome to endoplasmic reticulum nucleus and to the Golgi apparatus is actually known as a endo-membrane system. Endo-membrane system. And the main functionality of it is to create proteins. And that's a really vital endomembrane system, and it's really vital to the functionality of our cells and our bodies. Now, let's move on to some of the fun organelles, and everybody's favorite, the mitochondria. Mitochondria. Everybody knows it to be the powerhouse of the cell, but if you write that on a test, you won't exactly get full points, because it's not very specific. Really, its functionality, of course, is to create ATP. And it's the site where most ATP is created, or adenosine triphosphate, which is the energy currency of the cell. And this ATP is created through cellular respiration. This is a very intricate process that we have to definitely go over in another video. And the main parts of cellular respiration that happen in the mitochondria are known as the citric acid cycle, and the electron transport chain, which is commonly abbreviated as the ETC. Now, looking at the shape of mitochondria and its structure, the main thing that we have to know as scientists and as biologists is that the inner part of the mitochondria with this S-like shape is known as the mitochondrial matrix. And adding to that, the mitochondria is actually made up of two membranes, much like the nucleus that we have in the middle of the cell, and the space in between the two membranes is known as the intermembrane space. Moving on, let's just go clockwise. And the next organelle that we have is the lysosome. The lysosome. Of course, the lysosome doesn't actually have a star in the organelle, but it's more of a spherical shape, and it's easy to differentiate if we have some sort of identifier, in this case, a star, to identify if it's a lysosome or not. And its main functionality is to break down molecules in the cell. And it does this through a process called hydrolysis. What exactly is hydrolysis? Well, we can imagine it like the opposite of dehydration synthesis. What is dehydration synthesis? Well, it's essentially when you have a combination or a polymer of certain substance. For example, let's just take proteins right here. We have two different chains of proteins. At the ends of these proteins, you usually have an OH and an H group. This is known as a hydroxyl group, and this is known as, well, a hydrogen. What happens in dehydration synthesis is that we actually connect these two chains, in this case of proteins, and release water. So we take this HO or OH and an H, and we turn it into H2O. Thereby, we also connect these protein structures. Yet, on the opposite, hydrolysis is the use of water to break up the protein chain. Well, in this case, it's proteins. For example, if we have a bunch of small little proteins here connected, and we actually want to insert a water molecule, we do so, and in the process, we actually create two separate chains. And it looks something like this. Important to note is the biochemistry and the names of these biochemical groups. The OH group is commonly known as a hydroxyl. And as you get further along in biology, it becomes more prominent to know how it really connects to the functionality of organisms and cells. But for now, as the pre-AP biology level, it's not necessary to understand. The main thing we have to understand, however, is the lysosome and its use of hydrolysis, or adding water to a protein chain or a polymer, can actually separate the chain or polymer into two different subsets. But when we talk about lysosomes, it's also important to talk about the peroxisome. The peroxisome. The peroxisome is, well, the site of chemical reaction. The site of chemical reaction. And its main purpose is, well, to carry out these chemical reactions. Usually, it could either break something or make a certain molecule. And in the process, the product usually always contains hydrogen peroxide, H2O2. But because of its state, this hydrogen peroxide commonly just turns into water. So the main byproduct of the peroxisome is water. That's mainly all you need to know. And another thing to add, just like the lysosome, it doesn't actually have a triangle. It's just easier to differentiate and identify through that. So when we look at the peroxisome, that's, well, the simplest organelle that you have to know. It's not asked about too much, and its main function is simply a site of chemical reaction. Now, we've gone over half the main organelles in the cell. And looking at the peroxisome, lysosome, mitochondria, the nucleus, endoplasmic reticulum, the endo-membrane system, well, we can look for a part 2 to look at the rest of the organelles that we need to know for pre-AP biology.